Hi, good afternoon, everyone. We are going to start in a few minutes. Want to give a few seconds for those on Zoom to join us. Very quickly, my name is Ginny Van Horn, and I'm a project manager with Innovation Ventures here at Cincinnati Children's. And I am so excited because we have two fantastic guests with us today. But I want to go over a few housekeeping items. If you are on Zoom, I will be monitoring the chat. Pose your questions in the chat, and I will then raise them with our speakers at the appropriate time. If you're here in the research auditorium, so wonderful to see your faces in person. At the appropriate time, just raise your hand. I can come over with a microphone, and you can pose your question. So for the introductions, first, I am so pleased to introduce Dr. Enrique Linschow of BridgeBio Pharma. He is a trained biophysicist and molecular biologist with more than 12 years of experience in research spanning genetic and epigenetic mechanisms in disease, genome editing, cancer immunotherapy, drug discovery, and technology development. Dr. Linschow is part of the asset acquisition team at BridgeBio, charged with finding, evaluating, and moving therapies from the bench to patients. Also, I have my colleague, Justin Levy, Director of Portfolio Management for Innovation Ventures. Justin leads a team to develop and implement commercialization strategies here at Cincinnati Children's that can have an impact on patients, children, and hospitals. So I hope everyone please join me in a warm welcome for our speakers. And first, I'm going to turn it over to Dr. Linschow.
Hi, everyone. Thank you for that introduction, Jeannie. And I'm so excited to be here in person. My first in-person presentation since the pandemic. So very exciting. As Jeannie mentioned, I'm a trained scientist. I did my PhD at the University of Pennsylvania, working in epigenetics with Shelley Berger. And then I did a postdoc with Jennifer Doudna, working on CRISPR genome editing. And I've been with BridgeBio since last year, working in their asset acquisition team. And I think a lot about how a lot of times as scientists working in academia, there isn't clear ways of translating what you're doing at the bench into therapies and actual technologies that can improve patients' lives. And that's the reason I decided to come to BridgeBio, and also why I'm so excited to be here today to talk a little bit about some of that translation.
Or you can use the keyboard.
It seems to be stuck. Let me see. Oh, I think it's... Okay, there we go. So we'll start today talking a little bit about the interest of BridgeBio, how translation happens, and then we'll have Justin here talk a little bit about potential exit points of technologies from Cincinnati, resources that you have here. It's a great place, and the office is really awesome from everything I've seen today and before. We'll talk a little bit about Innovation Ventures case studies, and then some case studies from BridgeBio. We'll also talk about why IP is important, how and when to disclose. And these are questions that I, as an academic, when I was a postdoc, I thought a little bit about, but I had no idea about when was the appropriate time. So I think these are really cool questions to think about at the moment. But I wanted to start here with this really interesting graph showing that more and more genetic diseases are being discovered. And a lot of this has been through technology and new technologies. We can see here, for example, in about 20 years ago, the first draft of the human genome was finished. And then if you've been following the papers this year, we've been able to finish even the centromere this year, which is really exciting, as well as repetitive sequences. And a lot of this was aided by high-throughput sequencing, next-generation sequencing that occurred around 2005. And since then, we have a lot of this really cool resources like the UK Biobank recruitment that started in 2006, Genome-Wide Association Studies Results Catalog initiated around 2009, ClinVar launches in around 2014, and then we've had more than 1 million Genome-Wide Association samples sequenced to date. And what's really cool here is if you look at this blue line here, these are the hundreds of monogenic disease-causing variants that have been discovered every year. We have about 4,000 genes that have been discovered to be important in monogenic diseases, but we also know that there's 20,000 genes or 20 to 25,000 genes in the human genome. So we're discovering more and more. This is really exciting. And then from the GWAS side, we've seen there's thousands of common disease GWAS associations that have been discovered as well up to date. And this is growing exponentially. And I think what's also really interesting here is that there is a huge unmet need in terms of genetic diseases. If we look at the most common diseases here, some of the ones you might be familiar with, heart failure, 7 million people in the US, Alzheimer's, 5 million people, cancer, about 16 million people. But genetic diseases, we have over 27 million people in the US who have a genetic disease. Of this, about 50% are children, and we only have a disease approved for 5% of these genetic diseases. And even if you look here at the cancers, 60% of the cancers are driven by genetics as well. So there's still a very giant unmet need, and this is really where BridgeBio comes in, hope through rigorous science. Our mission is really to discover, create, test, and deliver transformative medicines to treat patients who suffer from genetic diseases and cancers with clear genetic drivers. We started in 2015, and since 2017, we're a publicly held biotech company. We have really fantastic leadership. Here we have our CEO, Neil Kumar, who came from Third Rock. We also have our investment committee, which is composed of really big veterans in the drug discovery and drug development field: Charles Holmesy, Frank McCormick, Richard Scheller, Phil Riley. And we have a really experienced research team that does really fantastic medicinal chemistry. For example, Robert Zamboni, Eli Wallace, Pedro Eltran, who have really good experience from Merck, Amgen. And then we also have a fantastic team that does gene therapy. And we leverage genetics to find therapies. So because we think that when the genetic driver of a disease is known, patients can be molecularly matched to therapy. And we really try to understand the genetic source of a disease to be able to understand what is the best way to target that disease and what can we do at the molecular level to treat that disease. And so far, our model has been fairly successful. We have 2 FDA-approved drugs. We have over 20 drugs in clinical trial and over 30 programs ongoing. So the way we find or look for new partners is we have the different models. For example, one of them is working with university business development offices like CCIV. We are in regular contact with over 50 academic institutions. We have over 20 academic collaborations. We often engage in scientific discussions with different PIs to learn specifically about the translational potential of their research. And we also receive inbound science through our network. And then we also do on-site or virtual workshops like the one we're doing today, where we introduce our model to academics and their institutions, and we provide industry perspective for researchers in one-on-one meetings as well. So if you're interested in collaborating with us, you can find out more about partnering with BridgeBio through my email here, enrique.linchao@bridgebio.com. You can also find me on LinkedIn, and we also welcome you to reach out directly through CCIV. Oh, okay. So, sorry, I moved the slides a little bit. And one of the things I really wanted to talk about today actually is the cost of drug development. And then that's something that when you are a researcher in academia, you don't think too much about, or you also are not familiar with. But on average, it costs between $1 and $2 billion to take a drug to market, which for me, the first time I heard this, I was shocked. And then the reason this costs so much and where academics usually are thinking about their work is up here, where we're thinking about a disease hypothesis and target discovery. And this can be also very costly. It can go from $0 to $1 billion in terms of being able to define the disease hypothesis and the target. But then after that, we expect to spend about $1 to $2 billion taking the hit target to hit, hit to lead, lead optimization, where you're thinking a lot about, for example, a small molecule, you have to make it soluble, you want to make it orally bioavailable, you want to make sure that it's not toxic, and you have to do a lot of testing in terms of, for example, preclinical models as well as animal studies. And this all can take several years. As you can see here on this line here, it can take about almost 5 years to complete this part. And then when you take this to phase 1, phase 2, phase 3, that can add another 5 to 6 or 7 years. And even after that, there's a long process before you launch. So this is really something that we also don't think about much, is that only 1 in 5,000 drugs actually make it to market. So you can think about all these development costs that have to go into this, and often you're not able to take your product to market, so you don't recover those costs. And then here we have the probability of technical and regulatory success. And this is really based on a lot of studies from economists looking at drug development in the past 20 years and looking at, okay, once we have a hypothesis, what is the probability of success for each stage? And you can see there's different probabilities here. Once you've identified the hypothesis, target to hit, you have a pretty high probability of success. Hit to lead, you start losing a little bit. But lead optimization, usually you have a pretty high probability of success, as well as reaching IND-enabling studies. And then it really depends on the indication that you're looking at. If you're looking at metabolic and derm, you have a pretty high probability of success in terms of completing phase 1 through 3. But if you're looking at indications like non-orphan metabolic, renal, second plus indications, you can see that phase 2 can be really low probability of success. So these are all based on numbers from existing programs, and you can see that it's very variable. All right.
Thanks, Enrique. Yeah, it's a pleasure to have you here. It's a pleasure to be here and talking to all of you. I wanted to talk a little bit about transitioning over to what it is to work with us at CCIV. The whole premise that we have at CCIV is on collaborating with you all together. This is a partnership between our office and you as the investigators. Really, this is about you coming up with your ideas and working with us to figure out who is the right folks to build relationships with, and talking through who are the right partners. And so the real key focus that we have is figuring out with you in partnership what is the right way to get these ideas into the market, into the hands of people that can actually get this into clinical development. And it's important to note that research, or if you're a clinician, institutional experiences might lead to new ideas that can benefit the public, but in many cases, publication alone isn't going to ensure that that's going to reach the market. And so that's where we're building in these partnerships that can come in. And we partner with those organizations to make that happen. And I want to talk a little bit about this sort of like funding gap, right? So, hearkening back to what Enrique was talking about with regards to that development path, right? There are these steps that we can think about development, right? And, here on the early side, maybe you have academic research funding that will help you discover these things. And here in the end, when we're at the late stage, we're going to have the commercial folks that are building it in. But really what we need to do is figure out how are we going to be able to get this stuff funded to make it attractive to someone who's developing those technologies. That's how we sort of figure out how do we attract a partner to take it on that might be willing to invest in these later stage studies. So this is where the partnering zone happens, and this is when we're really working together to figure out, okay, what is your technology? What are the things that we need to figure out? Who is the right partner depending on the stage that you're at to figure out how do we partner with those folks? And this, again, harkens back to what Enrique was talking about with regards to stage of development. He's going through that funnel where he's talking about that 5-year development pipeline. That's actually about right when we're talking about when we get a disclosure from you all, we're getting that early stage disclosure. Maybe you've just published on the mechanism. Maybe you have some tool compound. In order to get to a place where we're actually finding that partner to develop it, it's going to take about 5 years or 6 years to actually find that partner. So it's important that when we're working together, we know this is, we're in this together. We're in this for the long haul to try to find that. And there are lots of things that make something that could, might not be successful, but the idea is that if we work together, we have a higher likelihood of finding that partner on the back end. That said, we also have, and thanks to a lot of Ginny's work developing broad-based partnerships, we have a lot of opportunities for early-stage partnerships to try to get us through that hump of figuring out how do we get dollars and resources to that mid-stage of development where we not necessarily have a partner yet. And this is just an example of the kind of partnerships that we have. Obviously, Enrique is here from BridgeBio. That's a really valued partnership that we have, and we'll talk a little bit about. They might even be willing to come even earlier than what we were talking about earlier, which is actually really speaks to the value of the partnership with Bridge, and one of the reasons we're so lucky to have them here. But in addition to that, right, we work with a lot of different partners on open innovation that focus on other indications. So BioMarin also focuses on genetic-based diseases, but we work with Pfizer, that have their own specializations. As you all may know, a lot of the big pharma companies might shift their interests from time to time, but Ginny is really fantastic at figuring out, okay, at any given moment, what is their interest and what are the right technologies that we have here to partner with them. I will say one of the benefits of working with Bridge is that their interests have been pretty similar over the course of their life cycle. So we kind of know when we're working with Bridge what's going to be in their wheelhouse versus what's not. I also want to highlight a little bit here venture partnerships, right? So this is a little bit of a different model than some of our other sort of established players. The venture partnerships, right, there are these entities here in town and partnerships that we've developed nationally that focus on investing in technologies through a startup mechanism. And that is another way that we can get an early stage technology out, where maybe we can do some of that development here through some of our funding, like our innovation fund, and our technology validation startup fund, where we can actually, as an institution, provide some of that investment to get through a hump, and that makes it attractive to actually form a startup around it and then have one of these investment partners create a new entity and startup. So that's another path that exists. I also want to put in a plug for our translational core labs. For anyone thinking about gene therapies, this is, I mean, I'm sure you all know about it, but it's a really fantastic resource. They do a lot of really great work and have helped a lot of our technology. So this is just a sampling of our established programs, but whenever we chat, we're always looking for who are the partners that maybe we don't have yet that might be a good fit for the technology, and figuring out how can we bring them in and get feedback on the technology to advance it forward. And I want to give a quick case study of what that looks like, and also give you a sense of the timeline, right? So Dr. Zhang, Dr. Tan disclosed to us a novel neuro-based vaccine platform in 2008 based on NIH federal funding, right? They had worked on this through continuous improvements, and this is actually a common theme that we see with technologies. A lot of times we'll get a technology early on, and really through additional iterations, right, there's continual improvements, and that actually shows that there's continued interest from the faculty, and it also allows us to keep potential partners engaged over time. It allows us to show, oh, we have something new, will you take a look at this? And it actually can create a build partnership that way. So through those continuous improvements and next generation developments from other funding sources, there were additional disclosures in 2012, 2017, and 2020. We actually had an initial agreement with a large pharma company and a company in a foreign market, but the large pharma company chose not to develop. So sometimes there's this like misstep that happens. Maybe we find a partner and they change strategic directions and it returns to us and we have to sort of figure it out another way. Once it came back to us, we were able to sort of invest some of our internal funds, the innovation fund, to show in vivo proof of concept starting in 2017, and that resulted in a license to a small local company in 2021, which was actually a founded company by one of our local investment seed entities, CincyTech. So again, this idea that, open to figuring out, do we license this to large pharma? If that's not the right opportunity, is this something that we can maybe do some funding internally and then work with one of our seed partners to fund it that way? We also, through those seed investors, we also do some of our startups. One of the technologies here I'll highlight is Aerovant, right? So this is a technology we did in partnership with Roivant, sort of a large investment entity that was based on, again, through a lot of innovation fund funding and also a collaboration with our translational core to develop the lentiviral gene therapy that ended up getting licensed out. That was Dr. Malik's sickle cell gene therapy, if anyone's heard the story there. But as you can see, we do this in all different asset classes, and so we're always open for those kinds of collaborations. Enrique, do you want to jump in here and sort of,
Yeah, so when we're thinking about when to come in, BridgeBio really can come in at a lot of different stages. Often, most of the assets that we've been looking at, we come in here after proof of concept and validation. But we're also open to go even earlier to discovery and concept or proof of concept. And only last year, we started BridgeBio X, which is our discovery side of BridgeBio, where we can bring in programs that are really, really early and taken into proof of concept, and then from there on. We're also open to assets that have passed clinical trial or are in phase 1 and phase 2. And it really depends on the program itself, where we invest in. Talking a little bit about our investment model is we usually, when we find a therapy that we think is exciting, for example, in academia, we'll create a startup or a company around that specific asset, and we'll bring it under the umbrella of BridgeBio. So you'll have access to medicinal chemists, you'll have access to regulatory folks, in a way that the researchers that come in with that program can focus solely on the science, while we'll do all the regulatory parts of it. And then once we commit to a program, we commit to bring it to IND. So a lot of times with larger pharma, what you'll see is sometimes they'll license a program, as Justin mentioned, and sometimes that program can get shelved. For us, if we're committed to this program and we bring it in, we'll take it to IND at least. And then I wanted to talk about 3 examples here of BridgeBio, of where we've worked with academia in the past, and then to give you a sense of some of the programs that we collaborate with. We are disease and modality agnostic, so we're open to almost any disease as long as there's a clear genetic pathway that we can target and where the source of the disease is clearly understood. And then in terms of modalities, we've invested in small molecules, gene therapy, as well as ASO and enzyme replacement. Okay, so we'll start here with Nulibry. So Nulibry is a CPMP replacement disease for molybdenum cofactor deficiency, type A. And one of the things that really characterizes BridgeBio is that no disease is too rare for us. As you can see here, the prevalence of this disease is about 100 people, both in the US and the EU. In terms of the pathophysiology, a lack of CPMP is needed to produce, CPMP is needed to produce molybdenum cofactor. And a lack of this leads to neurotoxic levels of sulfite that cause CNS injuries and seizures. You can see here a couple of patients, for example. Children with this disease are living with sulfite intoxication. So here we know that the genetic driver is loss of function of the MLCS1 gene. And our therapeutic hypothesis was that when we added synthetic CPMP, we could increase molybdenum cofactor to levels that then decrease sulfite. And that way we could target the disease at the source and cure the disease. This has actually led to the first approved therapy for MLCD type A. We see 80% reduction in mortality, and we see improvements of the likelihood of developmental progression. Here we have an example of a survivability probability curve, and we can see that compared to the natural history of patients where you see a decreased survivability with increasing months, Nulibry shows a very steady state. And in these studies, Nulibry reduced the risk of death by 82% compared to untreated genotype match natural history controls. And then we were able to get FDA approval in February of 2021, last year. It's the first drug that we approved from BridgeBio, so very exciting. We received priority review. This was granted a label that allows treatment of presumptive patients as well. Origin is the company that we created around this asset for the US and Europe. It also has rare pediatric disease designation and breakthrough therapy designation in the US. And we also have a partnership with Sentinel Therapeutics for Nulibry for expanded market access. Here we have another example, limb-girdle muscular dystrophy. Here the prevalence is about 7,000 in both the US and the EU. The pathophysiology is we observe progressive muscle weakness resulting in a loss of ability to perform routine daily functions. Often patients have to be in a wheelchair around their 20s. And here the genetic driver is loss of function of FKRP gene. The therapeutic hypothesis here was that if we could add glycosylation substrate, we could drive residual enzyme activity. So far, what we've seen is that this naturally occurring compound that we're using is strong and safe. This is the first potential disease-targeting therapy that we've observed, and we also have oral dosing. And this is really exciting. We can see that if KRP mutations, what has been observed is that the vast majority of patients with these mutations have a homozygous double mutation in L2761I. And what we can see is in this case, patients are asymptomatic until late childhood, where they start to present with symptoms like lower limb and proximal weakness, hypertrophy of calves. And then by age 40, 25% of them are non-ambulatory. You see respiratory decline, non-invasive assistance is required by 10% by age 40, and invasive assistance by 1% of patients. And you also see cardiac dysfunction in about 10% by age 30. And then what we also observe is there's heterozygotes that have both this mutation as well as a second mutation. And this can be more severe, where patients are asymptomatic at first, but then you can already see symptoms at early childhood, like lower limb and proximal weakness, calf hypertrophy. And then most patients are non-ambulatory by age 20. There's respiratory decline. Invasive assistance is required by age 30 by 5% of patients. 25% of patients have cardiac dysfunction. So here in terms of the science, what we know is functional FKRP is required to fully glycosylate alpha-dystroglycan, which stabilizes cells by binding extracellular ligands. In LGMD2I disease, we see that partial loss of function through mutations in FKRP result in dysfunctional hypoglycosylated alpha-dystroglycan, which increases cell susceptibility to damage, as we can observe here in this process. So our therapeutic approach here was to supply supraphysiological levels of ribitol, a sugar, upstream to drive, and this should drive residual activity of mutant FKRP enzyme and increase alpha-dystroglycan glycosylation levels. And that's what we observe. We've been able to restore alpha-dystroglycan levels in cardiac and skeletal muscles. We see decreased muscle pathology through 6 months of treatment. We see improved respiratory function as assessed by expired volume and minute volume. And we also see increased running distance and length of running time. So this preclinical data has supported initiation of a phase 1 trial, and the first patients were dosed last year. So yeah, in phase 1, we were able to observe strong safety profile with all doses well tolerated. Dose was proportional exposures up to 15 grams. And this really informed our phase 2 dosing scheme where we're at right now. And then the last one I thought would be interesting to talk a little bit about is gene therapy using AAV9, especially since there's such a strong gene therapy core here at Cincinnati. So the prevalence of this disease is about 1,000 in the US and the EU. The pathophysiology is you see an inability to break down N-acetylaspartate and then NAA, which leads to demyelination, neurodegeneration, and loss of motor function. The genetic driver here is loss of function of the ASPA gene. And the therapeutic hypothesis here was using an AAV9, we could provide ASPA back to the patient. What we have observed is a profound phenotypic rescue shown in mouse models. We see delivery to the central nervous system by IV, AAV9, which has been validated. And we have a clear marker, NAA, to measure effect of therapy. In terms of the epidemiology and pathophysiology, this is an ultra-rare fatal autosomal recessive leukodystrophy. It's caused by biallelic loss of function mutations in aspartoacylase gene, ASPA. And then this deficiency prevents breakdown of N-acetylaspartate into aspartate and acetate, which results in failure to develop and maintain myelination in brain. Onset is early, around 6 months of age. We see profound neurodevelopmental delay. We see global cognitive language and motor impairment, poor head control, dystonia, seizures, and spasticity. And many patients die by age 10. Very few survive beyond. Care is supportive right now, palliative, no approved treatments. So here we actually work with an academic, Guangping Cao. our scientific founder. He has developed 3 generations of optimized HASPA transgenes, all packaged in AAV9 with a CB6 promoter. BBP812 is the vector with the 3rd generation transgene that we're using. Vectors were injected into ASPA knockout mice, which have a short lifespan because of hydrocephaly and then aciduria, extensive white matter evacuation, motor dysfunction. And then in terms of results, we observed that untreated mice had poor weight gain and died at 4 weeks, while mice treated with the 3rd generation vector had fully normal wild-type lifespan and weight gain. Treated mice had normalized ASPA expression and A levels, and the brains of treated mice were indistinguishable from wild-type, while untreated mice had severe vacuolization of the CNS. And then mice treated with the 3rd generation vector outperformed wild-type mice on a rotarod study. And a lot of this work was done with BridgeBio after this had been discovered. And then, yeah, I think what I want to mention here is that we've dosed our first patients this year with this gene therapy. So we're really excited to see where this goes. Back to you.
So, you've kind of seen an overview of what overall development looks like. You've sort of seen what it looks like sort of in the broad sense of what it looks like us, and now you've seen some examples. So the thing that I want to transition to now is what is it the initial stages of working with us? And I want to start from the IP perspective, because this is a really, really important point to get someone like Bridge or another entity interested. And I have a very simple diagram here with kind of a very complicated citation. And the reason for that really is that there is a lot of debate sort of when intellectual property is necessary, right? And there's a lot of studies talking about this. But ultimately, you're really looking for this relative distinction of a high development cost versus low cost of copying. And you're welcome to look at these citations talking about people indicating when IP is or isn't necessary from a sort of a legal perspective. But it's really important to know that when this is true, when there's a relatively high development cost to a relatively low cost of copying, in order to get an entity to invest in those development, they really do need that IP, that exclusivity on the back end in order to justify that development. And if we can't get that IP, then it's going to be really hard to push that technology forward into development and into the hands of patients and clinicians. So that's one of the things that I really want to drive forward, is that we can have a conversation of what is the IP that is necessary that's really going to allow us to leverage a relationship and get an investment in, so that we can get it into the hands of patients and clinicians. And one of the things that I think is really important about when to disclose, right? So this is like, if there's one thing you remember from the entire presentation, and hopefully Enrique will back me up on this, it's going to be this, right? Publication or presentation before a patent filing usually results in the inability to ultimately get a patent. So we don't always need a patent, but if we do need a patent, you need to let us know before you publish or present. And that's a really, really important aspect, because if we miss that opportunity, then we're never going to be able to get Enrique or Bridge interested in the technology. So it's really important that you come talk to us before you're publishing or presenting. And I also just really want to highlight this idea of a presentation. That in itself can be tricky. What isn't or isn't a public presentation, right? Here in the research auditorium, although this is at a children's facility, right, people can walk in, and this could constitute what's called a public disclosure in the patent space. And so even a presentation here in the research auditorium could constitute a public disclosure that could make a patent hard to get. So it's really important that we talk before any kind of presentation, any kind of publication. So before you do that, you can disclose it to us. And just for clarity, we're never going to tell you not to publish or present. Right? It's really important from our perspective. We know that you need to publish and present in order to continue your career, to continue to work. We're not going to stop that, right? All we're asking is that you let us know before that happens. So if we need to file, we have the opportunity to do so. And so the ask is to disclose it to us. And we'll talk in a minute about disclosing. And so that is on our inventor portal here. This is a long link. It's available on our Centerlink page. It's available on our website. I think these slides will be available so you can access them later. If you ever email me, it's usually at the bottom of my signature, so we try to make it easy for you to find this link. And if you have trouble finding it, just email one of us and we'll send you the link and you can disclose the invention. In general, right, we'll acknowledge the receipt within 3 business days and then try to work with you to figure out, have that conversation on, is IP necessary? Do we need to file right now? And so ultimately, this is what the sort of long-term sort of collaboration with us looks like. You know, on our side, right, we're going to commit to looking at the IP, protecting the IP if necessary, looking at the market, the regulatory analysis to figure out what could that development strategy look like. And in particular, one of the ways that we do that is actually getting input from advisors in industry. So again, one of the benefits of a relationship with Bridge is that they're willing to come in and look at stuff, even if it might not end up in a relationship. We can call on some of these program partners and say, hey, can you look at this technology, give us input, give us advice on what that development strategy looks like. And that helps us even if they're not going to license it. Do we need to go to the innovation fund and figure out, okay, we need to fund this work, and this is the key next step to get someone interested. And that, again, goes in line with identifying what that next step is to get that to that commercialization endpoint. We also will develop marketing materials. So we'll go to trade shows and try to build relationships with folks over time. And so we'll develop one-pagers or maybe collect your publications in a resource that we can easily share with interested parties. And then we'll facilitate those conversations with it. So generally, when we'll approach someone with some materials, the next thing that they're going to do is talk to you. You're the expert. You know the technology best. We're not going to be able to represent it in the same way that you can. So the next thing is going to be getting you on the line, having that conversation. And that's one of the things that I think it's important to acknowledge. This is a partnership, right? We might be able to tell you what the next development steps are, but we can't. But you know, you're going to need to sort of help us in figuring out, okay, what is the right funding source? Can you apply for that funding source to do it? Are you willing to continue to sort of work on those milestones if we can find the right funding sources and partners? And also, if we're filing IP, unfortunately, patent prosecution is a very deadline-driven process. And so we're going to need your help in making sure that we're responding to the patent office as well to make sure that we're responding to whatever requests they have. It's not in this presentation. We have another presentation coming up that's actually going to go through the patent process, so I'd encourage you to attend that to learn all about that. But ultimately, what you need to know is that we're going to need your feedback on an ongoing basis. And then finally, being available to have those conversations with us and those potential partners, because that's really what's going to breed success. And more, and Enrique, you can confirm this if you like, even after we license the technology, more and more, there needs to be that ongoing relationship even after the license, right? I think it used to be that we would be able to license a technology and, we'd give them the patent and then they'd be able to go off and develop it. But now more and more technologies have gotten so complicated that really the expertise that you have is necessary even after the license. And a lot of times there might be some kind of sponsored research arrangement back into the lab from the licensed technology. So this is something, not only do we need your partnership while we're trying to find the partner, but even after the license, there's going to be that ongoing conversation. And this is actually what I was talking about on May 12th. We are going to have an IP-specific process. So if you're interested in learning about what does that whole patent process look like, what does the timing of that look like, that's a whole nother conversation. That is going to be coming up on May 12th. And then I mentioned the Innovation Fund a couple of times, which is, again, our internal gap funding program. So our next round of Innovation Fund is September 1, and so we're going to have an informational session on that July 14th. And then I think we're also going to have it's not on here, but I think there's going to be a sort of a new faculty orientation conversation in August, I believe. So keep an eye out for that as well. If you have new faculty that you're aware of and they want to sort of get an overview, feel free to reach out to us and we can give you information on signing up for that. I'm going to admit Dr. Wu here.
All right. I wanted to pipe in really quickly. A couple of you asked what CCIV is. It's Cincinnati Children's Innovation Ventures. And so people have to get acclimated to our acronym, so apologies about that, how we use it. So Innovation Ventures, we're definitely part of Cincinnati Children's. A couple of you asked the Innovation Fund, as Justin noted, it's an internal pre-seed fund we have to help enable product development and commercialization. You will see flyers up in the elevator soon, but that's July 14th at 9, an information session. Letters of intent will be due September 1. And the May 12th, we're really excited. Our new patent manager, Ali Kruger, is hosting an event on Zoom talking all about the IP process from soup to nuts. It's going to be great. Flyers again in the elevator. You can always send me an email, send Justin, we'll get information to you. Now, gentlemen, we do have questions. So hold on. First one, and I don't know which one of you wants to tackle this, but for such rare diseases, how do you justify funding? I mean, do you feel like you have to get recruitment for clinical trials? And I don't know, Enrique, if that's something you would like to tackle.
Yeah, I'd say for
Yep, turn it on.
okay. Hello? Oh, yeah. So the model that we use at BridgeBio is that if we have a diversified portfolio, if it's diversified enough, then we can cover also ultra-rare diseases and ultra-rare genetic diseases, even if that doesn't lead to any commercial value. We think it's really important that there's a system that allows for development of those drugs as well. So our strategy is having a diversified portfolio.
That's loud. I also want to add on to that. Let me see if I can find the right distance. Hello? Hello? I want to add on to that, in that a lot of times an ultra-rare disease, so Bridge is a great partner in that it might be eligible for their partnership. If Bridge isn't the right fit because it's not the right indication or it's not genetically driven or whatever, a lot of times an ultra-rare indication can be the lead-in to a platform technology that could actually be, so that ultra-rare disease could prove as a proof of concept that then can scale to other technologies. So we actually just executed a license with a young company based on an ultra-rare disease that's being sort of proved out here, and the sort of delivery platform can be used for sort of broader lung diseases. And so the entity that we license to is interested in that platform. That ultra-rare disease operates as the proof of concept, and then that can be leveraged for broader diseases. So there are strategies that we can use if you're working in an ultra-rare disease. It just depends on whether there's an entity like Bridge that's interested or if it can be leveraged for a platform.
Great, thank you. Do you have a follow-on?
Yeah, I just wanted to add that for a lot of rare diseases, there's also patient foundations who have been able to fund a lot of these studies and also interested in taking a lot of the therapies to commercial stage. So they're always a good partner. That does open a lot of questions about equity and some diseases that are underfunded. Yes, that's an ongoing problem as well.
Great. Another question from the Zoom chat. It seems the pattern with industry is to provide funding to academics, which is great, all right? However, there are some resources that are scarce in academia, like biophysical characterization, screening compounds, assay miniaturization assistance. Have you ever considered including those types of resources in addition to funding?
I would say, at least for BridgeBio, if we have a very strong therapeutic hypothesis, we do fund a lot of those characterizations, CRO. We also take compounds from from target to hit and also to lead. So you often don't really need to have all of that done prior to approaching BridgeBio. We are open to taking things that are very early discovery all the way, and then often we want to do the lead optimization ourselves, because there's always going to be parts that you want to optimize further. But what we really want to see in terms of the data is a clear hypothesis as well as a clear way to connect the dots back to the disease and back to the therapy and how that therapy is going to help in that disease specifically.
It's such a great question because, you know, it's just true that there are these gaps in development, right? And I think, you know, when we chat with a faculty member that is, you know, needs to miniaturize the assay, let's say, to, like, you know, do better screening. I mean, it's a challenge. I think that this is an ongoing conversation, and we know that these gaps exist. It's possible, right, in the way that Bridge might be willing to, you know, work with a CRO to get some of that stuff. That could be possible for an innovation fund support. So an innovation fund support isn't necessarily limited to funding for the lab. It could be used for funding to external parties to do some of that work. And so we're sort of aware of those gaps, and we're trying to work through strategies to do it like what we have with the innovation fund.
All right. We want to talk a little bit about IP, actually, but first let me try. What role does the Cincinnati Children's PI play as the product moves along the developmental pipeline?
So I can talk a little bit about our model at BridgeBio, and that really depends on the PI themselves. We have had PIs who want to be involved and others who just want their technology to be out-licensed and they don't want to have anything to do with it. For most of the people we've worked with, though, they want to be part of the next steps and they want to see their technology go the way through. And we're really open to that because we believe that you, as a PI, and the person who's bringing the technology to us are the experts in that specific technology. So we have different models. What we usually do is we bring in the PI as a co-founder of the entity, and then they can decide how much of their time they want to put into that. They don't have to be a CEO, they don't have to be a CSO, they don't even have to be in the C-suite. They can just be part of the board. They can provide some expertise through there, but we'll handle everything else. We'll handle operations, we'll handle all the regulatory parts, and we'll also bring scientists specifically for that asset as well. So it really depends on what you as a PI want to do and how much time you want to engage.
Yeah, totally agree with everything that Enrique said. The thing that I would add on from a sort of children's focused perspective is, generally there does need to be some technology transition from the institution to the licensing entity. And a lot of times with an entity like with Bridge would create or with a startup company, generally there's a scientific advisory board, and that's generally the vehicle through which the PI would remain involved. What I would say, and maybe I'd be curious to hear if this is true at Bridge, I find that PI is involved through some of that early stage development, but as the company really scales and gets into later stage development, sort of there's a different expertise that is utilized. And so there's that initial transition getting through some of the early stage development, and that's where the PI's expertise is really necessary. And then there comes a point where there's a different sort of level of expertise getting in through later stage trials and things like that. So that's what I would say.
Yeah, I would add to that for us, a lot of times the PIs come and stay all the way to IND. And then at that point, they can decide if they want to convert the percentage of ownership that they've received as shares and leave, or if they want to continue being part of the company and see it through clinical trials as well. So they have the option once we reach IND. And at that point, the company is also valued externally, so there's other incentives as well for the PI.
And I would just say, I think if, with regards to sort of the equity in the shares, Children's has an IP policy that allows distribution of revenue from Children's through the PI. That's kind of a longer conversation on what ownership looks like, how that acquires in general from the IP perspective, right? We license the IP and then revenue flows through the institution and then back to the investigator. But that's a sort of one-off individual conversation, so we'd be happy to engage on that if anyone has specific questions.
Well, you must have read this person's mind, because the question was, does Cincinnati Children's or the inventor have an ownership stake in the final therapeutic, and what does that look like?
Great question. So, yes. Really, it depends on who we're licensing the technology to. So if we're licensing to an established pharma company, right, we're not going to get public shares in Takeda, right, just as an example, if we were to license a technology to Takeda, right? So there's different sort of ways to collect revenue and value from the technologies that we license, regardless of how we collect that revenue, whether it's equity in a young company, whether it's cash, whether it's usually we'll have a term around a percentage of net sales based on the sales of the product. There might be milestone fees. There's a whole slew of financial negotiations that happen that we sort of work on through our office. But whenever the revenue comes back into the institution, and I'm happy to we can share our IP policy. This is all in our IP policy, which is available on our Centerlink page. A certain percentage goes to the research foundation, a certain percentage goes to the division from where the invention came from, and a certain percentage actually goes to the PI themselves, and they have the opportunity to keep that or actually redirect the funds to their lab here at Children's. That's our policy. Happy to answer more specifics on that, but it might be better for one-on-one, but I don't know if you want to add.
Yeah, I think at additionally to that, if the PI decides to be part of this company and become a founder, there'll be a percentage of non-dilutable equity that will go to that PI, and that will become non-dilutable up to the point of IND. And then after that, the PI can decide if they want to leave with that percentage or if they want to let those shares convert to BridgeBio shares as well, and then they can continue being part of the company as well.
Yeah, and I just want to add, so I would encourage, if this is of interest to you, to have one-on-one conversations with us, because the founders piece can be complicated. One of the things that we need to sort of work through is our potential conflicts of interest, and as a result of that, in general, being a founder can create other complexities. And so there are ways to be involved in the company that allows for some flexibility, and then there are ways that makes more sense sometimes to come through the children's side. Again, I think that's a sort of different, deeper conversation, but happy to have one-on-one conversations given the specifics.
Thank you both. We have about 10 minutes. Are there any questions from anyone in the research auditorium? Yes.
Hi, sorry. My name is Ola. I'm from University of Cincinnati. So I had a question about the patent process, the difference between the provisional status and the conversion, and when we could publish between both of these. I believe there's like a year or so between, so.
Yeah, thanks for that question. So again, you should come to the May 12th conversation. We'll go into a lot more detail. So I'm reluctant to go into too much specifics because I'm going to say something and I don't want anyone to hang on it. You're correct that the U.S. does have a 1-year grace period between publication and like when we would need to file, right? However, that's just the U.S. And so a U.S. filing does not necessarily result in protection around the world. And in general, for an entity like Bridge or other entities, they will want at least European protection. And so if a publication happens and then we put a filing in, that could prevent European protection, which could endanger a potential licensing opportunity, which is why disclosure to us before publication is very important. Now, that said, in extenuating circumstances, we can figure out, okay, what strategy can we take on here? What do we see as iterations down the road that we can file improvements on? And there's, happy to have those one-on-one conversations. Now, you mentioned a provisional application. So in the U.S., there's something called a U.S. provisional application, which allows you to sort of get in a filing date. And then a year from that, you can file various different kinds of applications. The most common one we do is what's called a patent cooperation treaty application. And that sort of preserves international rights. And then after that, there's another 18 months where you can file in individual countries. In general, between the provisional stage and the PCT stage, you can add additional data into the specification into the PCT, but that becomes a little tricky because you only get protection of the filing date from what was in the provisional. So one thing that's risky sometimes is a PI will disclose to us. We'll file a provisional application on what was disclosed to us. The PI will then say, great, I have a filing application, now I can disclose whatever I want for the next year. You can only disclose what was put in the provisional because that's what we have the filing date on. So if you're disclosing on over that course of that year, you have to keep letting us know so we can update that provisional filing to protect on an ongoing basis. Hopefully that answered the question.
Yes, thank you.
All right. Here's one actually for Enrique. Do you have any opportunities for graduate students at BridgeBio?
I would say we don't currently have opportunities. We have in the past had internships, but I would encourage anyone who is a grad student to reach out to me directly as well. You can do it through LinkedIn. If you're thinking about careers outside of academia, I spend a lot of my time mentoring people who are thinking about alternate or so-called alternative careers. So I'm very happy to talk to any of you one-on-one.
Thank you both. Any final questions? We have just a few minutes remaining. Yes, Moo.
Oh, yeah, sorry.
So I guess I have two questions. So one thing that, Enrique, you have mentioned that you're looking for genetic diseases with clear genetic pathway. So one question related to that is, how do you go about finding different modalities? Are you modality agnostic, or there are specific kind of modalities that you're going to be interested in? And then the second question is that, there are over 7,000 different rare diseases with genetic disorders. So, in addition to clear genetic signatures, are there any key piece of data or information that you're looking to utilize as a selection criteria when you're thinking about partner with different PIs?
Yeah, I would say for the first question, we're modality agnostic. And in our pipeline, we have ASO, small molecules, gene therapy, enzyme replacement as well. So we're really open to any modality as long as we can target the source of the disease. We're also open to polygenic diseases as well as we can actually connect the dots and trace the source of the disease. And yeah, as you mentioned, there's over 7,000 genetic diseases. A lot of diseases, unfortunately, you cannot treat, or the treatment window might be really small. So those are hard to tackle. So we, in terms of the data that we're looking for, we often want to see, and this really depends on the disease and the different treatment option. We want to see some preclinical data. We want to see that the hypothesis that the PIs have is tractable. So if there's a lot of publications out there that really explain this is the source of the disease, there's genetic data out there showing that variations in that gene, for example, lead to the phenotypes. We understand penetrance of the disease. So the more evidence we can gather around a disease, the better it will be for us. And a lot of that work we also do in-house. We have computational geneticists. We can work together with the PI to complete some of that work. So for us, really, we encourage PIs to reach out to us as early as they can and see what we can complete and what we expect from their side as well.
All right.
Thank you.
So
Thank you.
everyone, thank you so much for joining us today, and please join me in extending a hearty thank you to Enrique Linshao and Justin Levy. If you have questions or you have ideas of upcoming seminars you want to see, please send me an email or send Justin an email that's up here on the slides, virginia.vanhorn with an E at cchmc.org, or Justin Levy at cchmc.org. We're here. We want to do new seminars and work with you, so don't hesitate to reach out. Also with BridgeBio, if you have an idea that's of interest, please don't hesitate to contact me, and we'll vet it with BridgeBio. So thanks again for your time, and we look forward to seeing you hopefully on May 12th at noon.